Okay, so I just wanted to start off with this little bit weird question. If you wanted to view something really, really tiny with a microscope, how would you do it? And the answer is right now it would be pretty hard. You probably, if you wanted to look at something at a greater magnifying magnitude than something like 10 to the like a 200x objective or something like that, you'd probably need a very powerful microscope or a scanning electron microscope or something like that. But if you wanted to measure the concentration of something at a really, really low concentration of individual molecules, you really can't do that on a existing typical stage microscope or dissecting microscope or something like that. So that leads us to this paper, which is called Single Molecule Detection on a Portable 3D Printed Microscope. And it's exactly what the title sounds like. So basically it detects individual molecules that you give to it, and the microscope itself is 3D printed. At least the housing is, and the code that's used to run it is also open source. And of course, because it is 3D printed, it is cheaper than a typical single molecule microscope. So before we get started, here are some quick definitions. So the first one is single molecule fluorescence. So again, that is what it sounds like. It's basically observing as opposed to a large batch of molecules or a large sample. It's looking at each individual molecule in the sample to determine its intensity or its concentration. And it's measuring that concentration by measuring the intensity of its fluorescence. Single photon counting goes along with the fluorescence. So when you hit something with a laser, it should fluoresce, or these, at least the proteins, they should fluoresce. And by counting the photons that are emitted from that fluorescent emission, you can determine the intensity and the concentration of the sample that you're studying. So fluorescence correlation spectroscopy is using that same principle of fluorescence emissions. The only differences here is we take the data and they run it through this very complicated algorithm that essentially generates a curve. It's a little bit hard to interpret, I'll show you later on, but basically that is the style of microscopy they're doing. All it means is they're running the data through a special algorithm that generates a curve. Confocal microscopy is pretty cool, so if you were to take a normal microscope, you would have to light the entire stage with a spotlight or the light underneath the stage, so everything would be lit. In confocal microscopy, this is the complete opposite. So it basically focuses the light through a really tiny pinhole, so only one tiny part of the sample is actually lit. And what that allows you to do is focus really, really well on individual parts of the sample. So you can focus on the entire sample by stitching together all the images you get, by stacking them, so you can get a really good 2D image where everything is in focus, or if you stack it properly, you can get a 3D image as well. And then the alpha-synuclein is in order to test this microscope, because they had built it themselves, they used this protein. It's associated with Alzheimer's disease. It does not cause Alzheimer's disease, but is related to the protein that does. But that's not really relevant. It's just a protein that they use to test it. And thioflavin T and Alexa 568 are two dyes. So the Alexa dye is the 568 refers to the laser. Like the wavelength of the laser that it'll fluoresce under, and thioflavin T, we'll get to later on, it basically, again, binds to alpha-synuclein and allows you to determine how it's fluorescing. And finally, a femtoliter, so you might have heard of microliters or milliliters, that's 10 to the -6th and 10 to the -3. You might have even heard of picoliters, which is 10 to the -12th. A femtoliter is 10 to the -15th. So these are really, really tiny concentrations. These are really tiny volumes, which is like you probably wouldn't be able to even see a femtoliter on a normal microscope, which is why you might use something like this to look at the individual molecules. So before we get to the experiments, here's just some background of why they did this. So a single molecule microscope is particularly useful if you're looking for biomarkers of disease, if you're looking for specific proteins in a small sample. They use it for liposomes later on. So these kinds of molecules that it's really hard that can be associated with disease, but it's kind of hard to do without the power of this kind of magnification. It's also useful if you want to look at biological processes at the cellular level and the molecular level as well. Right now, one of the reasons that this was developed, this specific microscope, is that it's mostly used in biophysics, but not in traditional biochemistry or structural biology, where it has a lot of applications. And that's mostly because it's pretty expensive and it requires a lot of training. Also, some of them require like dark rooms and things like that. If something goes wrong, you have to get a technician. If you want to collect, because this is all based on emissions, you have to be able to collect viable data sets, and that's really, really hard to do. But new developments have allowed this, so yes.
Just a quick question, you might not know the answer to this, but I've worked with a lot of different confocal microscopes, 2-photon microscopes. How do they get away with it not being in a dark room?
They have like, I'll show you, there's the next image.
Okay.
I'll get to it, but it has like a little, like a hatch kind of thing.
Like some kind of shield, okay.
Yeah. So the new developments that allow this to happen are super high resolution imaging. So we all have cool cameras on our phones. If we use that in a microscope at a higher resolution for scientific purposes, we can stack those images really clearly and see what you're looking at. As well as the ability to kind of squish things together. So this particular microscope is centered around a single photon counting molecule, which we'll get into later. But by the magic of technology, those things have been shrunk. So this molecule is pretty compact. And again, this doesn't need any specific environmental conditions. So the goal of the researchers was to observe molecules and processes cheaply and quickly, but also something that a science lab could use without going like the full, how many ever thousands of dollars it takes to buy a big one of these, or a school could use, just for educational purposes. So here they show how they built the microscope. So they sourced all these different parts. So the first is the laser. So the cool thing about this microscope is depending on what dye you use and what molecule you're using, you can swap out the laser for different wavelengths, which is helpful for the fluorescence. So all that is doing is it gets bounced through, it gets run through an ND filter, gets bounced through a mirror, hits the sample, which is going to be under the objective. Then that fluorescent emission gets bounced back out of the sample and runs into this. This is just a dichroic piece of glass, which means depending on how the light hits it, it'll output 2 different colors. But it hits this single photon counting molecule. So this is where the actual measurements happen. And it uses something called single photon avalanche counting, where it basically releases an avalanche current. And by measuring the leading edge of that current, that huge amount of current, you can determine when the photon was released, when the fluorescent emission happened, and determine the intensity and the concentration of the substance by doing that. B is the 3D printed housing they made. So they went through, I don't remember what CAD software they used, but they basically custom built this entirely based on the parts they're using. Obviously, this is pretty customizable. All their stuff is available online. And then this is the really cool part, is how small it is. So you can see here, like this is maybe like the size of a big pencil case or something. It's smaller than the laptop. It's not, plugged into any giant machines, just a few USB converters and things like that. And the software that they're using to run it is also, it's based on LabVIEW, which is a program, and they made some special code for it, which is also open source. So if you wanted to buy all these parts, and assemble it, you could totally do that. You have the CAD model here for the structure, you have the code here to view it, and you have all the parts that you need based on this diagram. So it is very user-friendly. So here we have the, basically their proof of concept figures. So D demonstrates basically the principle of confocal microscopy, how you're focusing the light onto one molecule, in this case it's fluorescing. E is, again, so you can see at the top they use 568 alpha synuclein, so that's that protein plus the dye. And with this, they compare it to a buffer to demonstrate that, yes, we can actually see something, and that the peaks on this are consistent in terms of intensity. In F, they used green fluorescent protein, so pretty common protein. The green curve is normal GFP, and the blue one is trimeric GFP. So basically, the point of this figure is that the trimeric GFP has a wider spread, which means there's more oligomerization, which you would expect. And then the inset is just they fit it to a Gaussian curve to amplify the difference between them. And in G, this is the correlation spectroscopy figure that I was talking about. It's pretty confusing to read, but basically it is showing that with no protein, some protein, and a protein plus a micelle, which mimics a membrane. So it's kind of like a lipid bilayer, but without the bilayer part, and not really but it's basically like a membrane. This demonstrates that you can clearly see the difference, and also this is a different wavelength if you see, it's a 488, so you can use it with different lasers, and it is able to differentiate between the different substances. Yes?
I just want to clarify, like, this paper is about making, like, a cheap, like, version of, like, a microscope, is that correct?
Yes, also one that's portable, and one that, like, a lab can use quickly, one that doesn't require a lot of training.
So, like, what's the price difference?
They don't actually go into it, and I didn't want to, but they did. I know they didn't use very fancy, like the 3D printing housing is made on like a typical like consumer printer, an Ultimaker. So like it's not, they use like standard filament and stuff like that, so it'll be like depending on how much, like what kind of because it is customizable depending on the configuration, but you know the price will obviously vary, but for some reason they didn't actually go into it. So here is another more their proof of concept kind of idea. So as you can see it shows thioflavin, which is again the dye binding to this amyloid fibril, which is the protein that they're using. And as the concentration increases, which you can see up here, as you would expect, the concentration, the observed intensity increases, so the microscope is detecting higher concentrations. Which is good. And the other really cool thing about it is as something as small as 1 nanomolar, which is 10 to the negative 9th, you can still see pretty consistent stuff, and even at the smaller ones there are a few peaks, so that kind of ability, the fact that you can see such small things at such a compact machine is pretty cool.
What's the and it's visualizing a really small area, right? This is intensity in one single area, whereas the sample are we moving, it's just intensity across time in one spot, yeah?
Yes.
Okay.
So this is a figure that basically demonstrates that this microscope is actually better than some existing methods. So if you look at the figure all the way on the left, the blue line is this microscope, this new one that they built, and the red line is something called fluorescent plate reader. So basically, it uses a large batch or a larger batch of your sample, and it uses again the fluorescent principle to determine like the concentration of it. But what you can see is that at really small volumes, between like 10 to the negative 12th and like 10 to the negative 6th, it can't really tell the difference between any of them. It thinks they're all at the same concentration because it's not that precise. And this is like a professional lab instrument. This is not like another homemade thing. Whereas the microscope that they developed is actually able to individually differentiate all these different concentrations. And it's obviously, if you're looking at things that are that small, you don't want to be thinking that 10 to the negative 6th, 10 to the negative 12th molarity is the same as 10 to the negative 6th. So this just demonstrates some of the limitations with this kind of microscopy. And then these two graphs, really quickly, are just deconvolutions of that one. So this demonstrates the number of fibrils that they're counting. And this one is the average intensity for the different concentrations. But the main thing here is that this is impressively more accurate. So in conclusion, the microscope is very novel because it is much more inexpensive. It's much smaller. Fluorescent plate readers are much larger. You can't just like plug it into a laptop and be like boom, like you can with this one. This is also self-calibrating. So I didn't mention that up here, but there's a mirror on a piezo motor. So what that allows you to do is if you're changing the laser wavelength or if you want to modify some parts, it's a motor, so you can adjust the way that mirror reflects the light. So that's pretty cool as well. So it's very customizable. You don't need to get like a lab technician to come in and figure out how to adjust that. You can do it yourself. It also does self-calibrate, so that it has a program that allows it to self-calibrate. So another bonus feature. There's no special training required to use it. You just plug it in, you use their software, their code. It can be 3D printed and assembled. Again, this isn't any like special 3D printing, it is just standard like plastic. And finally, it's really easily, it's more precise than the existing methods. So Again, we saw that it's better than fluorescent plate reading. And if you're doing this in like a lab, like just to test it out, see how it works, if you're doing it in a school, this is definitely a great option. And especially if it spurs the competition to shrink their respective microscopes. Thank you.